Hello students, this is Dr. Faizan Mirza. Here we are discussing how your taste receptors function. Taste receptors are sensory cells present on the tongue, and these are specialized cells which are located on the surface of the tongue, and not exactly on the surface of the tongue. The surface of the tongue is actually having these finger-like projections called papilla. You can probably just see these if you just try to look at your tongue using a mirror. And in the reflection, you can very well see these papilla. In between two adjacent papilla, there is a depression, there is a groove, and around that groove, around that depression, there are clusters of cells. If you magnify this cluster of cells, this is a taste bud. The taste bud here I'm discussing is composed of several cells here, and all cells are having these microvilli protruding out into this groove or this depression here. All these cells are the taste receptors, and together they form the taste bud. Now, the taste receptor that we are discussing here is the one which is designed for tasting salt, which is sodium chloride. This is the taste receptor cell. This is a sensory cell. This is not a sensory neuron. This is a sensory cell. The sensory cell makes a synapse with a sensory neuron, and this sensory neuron then takes the message to the central nervous system from your tongue to the brain, where you actually recognize what taste was there, and you just either continue eating it or you just puke out. Okay, let's continue. So this is a sensory cell here. On the surface of the sensory cell, these are the microvilli here. So let's just magnify this. So when the sensory cell is in a resting state, this cell is not permeable to sodium at all. We know that the cell is depolarized when the salt is not there. So when the cell is in a resting state, this will be a resting membrane potential inside the cell, and at this point there will be vesicles here which will be storing neurotransmitters, which will already be stored at this end of the sensory cell. Keep in mind that this end of the sensory cell is the one which is close to the sensory neuron where the synapse is being formed. Now what happens? Let's say you are taking food and there is sodium in your food, so that sodium chloride in your food triggers the entry of sodium into the cell. The entry of sodium, we know it depolarizes the cell membrane. So this cell initially was at resting membrane potential. It was at -70 all along, for example. So the entry of sodium caused localized depolarization from the resting membrane potential to the threshold value, which is from -70 to -55. At -55 millivolts, we know that voltage-gated sodium channels start to open and more sodium ions start to enter into the cell, depolarizing it fully. So the wave of depolarization that begins here start to spread all along the sensory cell. So every subsequent part of this sensory cell keeps getting depolarized because of the entry of sodium ion. A moment comes when the wave of depolarization reach this point in the cell membrane where there is a voltage-gated calcium channel located. In fact, there are voltage-gated calcium channels located, multiple of these. So the voltage-gated calcium channels open up because the voltage inside the cell is positive and this region is depolarized. So the opening of this voltage-gated calcium channel allows calcium ions to enter into the cell, into the sensory cell. Once the calcium ion are there, the calcium ion bind to these vesicles. They trigger the release of the neurotransmitter into the synaptic cleft here, and it binds to the next neuron and the sensory neuron, and this is how the stimulus just goes ahead. So this is how the membrane potential was negative initially in both the cells, and we have already high calcium outside the cell. This is always the case. And these are the vesicles which are having the neurotransmitters stored in your sensory cells. Now, when we know that the cell was at resting membrane potential. Now the cell got depolarized. As I told you, the wave of depolarization spreads until when it reaches this point, the calcium voltage-gated channels open up and calcium ions start to enter, and it triggers the release of these neurotransmitter vesicles. They undergo exocytosis, and this is released into the synaptic cleft. The release of these neurotransmitter triggers the next neuron, which is the sensory neuron, to get stimulated. This is how the neurotransmitter release these blue dots, and they cause the next cell to get depolarized. So this is how your taste receptors function. So we have generation of receptor potential. So receptor potential at times can be generated at the sensory cell. For example, the taste receptor. We have similar kind of sensory receptors, sensory cells present in our nose, which can bind to a certain chemical, and then they trigger the release of a neurotransmitter, which cause the sensory neuron to take the information to the brain, which then recognize that particular smell as being a good smell or a bad smell. Similarly, at other points in the body, there is no sensory cell, there is a sensory neuron ending. So this region is actually the ending of the sensory neuron. So there are two cases in which we have sensory information going to our central nervous system. either through a sensory cell sending the information or the impulse to the sensory neuron through this synapse, or the sensory neuron itself is having a sensory ending which is present in the body, which collects the information or the stimulus which receives the stimulus of a certain type from the body, either external or internal. So we know that this region, this is shaded green, is sensory zones in both the cells. So both the cells, for example, I'm discussing this setup only. So the same applies here as well. So this cell will be depolarized fully. It will be at resting membrane potential, having a negative potential all along because it's not getting any stimulus right now. So in the absence of the stimulus, so this cell is at resting potential. This is resting potential as well. They both will have minus 90 or minus 70 millivolt of resting membrane potential, which is a relatively negative environment inside the cell. This is the tissue fluid outside these both cells. So once the stimulus comes, this causes the cell to get depolarized. So this region becomes depolarized. The sensory zone got depolarized, because of which ion entering? The sodium ions entered. And they cause the membrane to depolarize from minus 70 to a value of threshold minus 55, at which the voltage gated sodium channels opened up and the value of potential difference turned from minus 55 to plus 30. So the membrane gets fully depolarized. So as the wave of depolarization spreads all along the cell, eventually voltage gated calcium channels start to open up, which are located only in this part of the cell where the neurotransmitters are stored in the vesicles. So the entry of these calcium ions, they trigger the release of this neurotransmitter, which is acetylcholine, into the synaptic cleft, into the gap which is present between these two cells. For example, I'm just mentioning this acetylcholine with this purple dot. So all these acetylcholine molecules are released here. The next neuron, which is the post-synaptic membrane, which is the sensory neuron here, this membrane is having these black dots. What are these black dots? These are AChR, acetylcholine receptors. So these purple dots, which are acetylcholine molecules, they can attach to the acetylcholine receptors and then cause the sensory neuron to get depolarized. For example, if this is the acetylcholine receptor, this is the inside of the cell, this is the outside of the cell, and these two yellow lines, they represent the cell membrane which is present here, and this black dot is the one which I have drawn. This is the acetylcholine receptor. This region O outside is the one where you have this purple dots of neurotransmitter released, and this I is inside this neuron, which is having a negative charge right now. So what happens now? We have acetylcholine molecules which are released in this region, in this gap. Acetylcholine molecules, they come and attach to the receptor. A single receptor allows two acetylcholine molecules to attach. When two acetylcholine molecules have attached here, a channel opens up in between this receptor, and this channel allows the entry of sodium ions into the cell. The entry of sodium ions into the cell depolarizes the membrane. Localized depolarization occurs from resting membrane potential to threshold. So this is entry of sodium triggering the potential difference to move from the resting membrane potential, which is minus 70 millivolt, to a threshold value of minus 55. So at minus 55, the voltage gated sodium channels, they quickly open up, and the cell becomes fully depolarized as more sodium keeps entering. So this is an example of a positive feedback. So sodium entered initially through this receptor channel, which got activated, which got opened up because of the two acetylcholine molecules attaching at one point. So one acetylcholine molecule sits here, the other sits here, and the channel opens up. So this sodium entry promotes the entry of more sodium through the voltage gated sodium channel. So there are two pathways through which sodium is entering into the cell. So first, the sodium enters through these receptor channels, which are opened because of two acetylcholine molecules attaching, and then the voltage gated sodium channels open up and they allow more sodium to enter. So this is an example of positive feedback. The membrane gets fully depolarized, and this is how the impulse just keeps going ahead to the central nervous system. So this is the post-synaptic neuron. It generates an impulse, and the impulse then is carried away. For example, now later on the stimulus ends. Now once the stimulus will end, this region, sensory zone, will gain repolarization. It will undergo recovery. When it undergoes recovery, it sends out potassium ions, and it regains its negative charge. We know that potassium efflux is actually causing the resting membrane potential to be set up once again. So the voltage gated calcium channels, they stop functioning. The calcium entry also stops. Then there is an enzyme, acetylcholine esterase. This acetylcholine esterase enzyme, it helps in degrading acetylcholine present in the synaptic cleft. So all these acetylcholine molecules, they are broken down into two pieces, or two parts. Acetyl and choline, both are separated from one another. Acetyl and choline are then transported back into the cell, so that they can be packaged once again and packed into the vesicle, so they can be released next time when required. Similarly, the next cell will also allow potassium ions to keep going out, and sodium entry will stop, and as a result, this cell will become repolarized as well. So this is how the pathway works all along the working of how the impulse is generated and how it is transferred. So it's always resting membrane potential, then threshold, then action potential, and then after action potential there is again RMP because of recovery. So RMP to action potential is always depolarization, and returning from action potential to back RMP is a repolarization. So that's it from my side. Thank you so much.